This animation is looking at the cell cycle. Let's start with a single cell. It's a new daughter cell of a cell division. It starts in the G1 phase, which is also known as the growth phase 1 or the gap phase 1. Within this cell, I've represented the nucleus in white, and inside the nucleus I've shown a single chromosome with a centromere. At this stage in the cell cycle, this chromosome consists of a single double-stranded piece of DNA. In other words, this chromosome has not yet replicated. Which leads us to the next phase, which is the synthesis phase of the cell cycle, or the S phase. This is where DNA replication occurs. During DNA replication, the DNA base pairs are separated and used as templates to create a complete copy of the original DNA. So at the end of the synthesis phase, you have a single chromosome, but now that chromosome has two sister chromatids, which is two pieces of double-stranded DNA that are identical to each other. The next stage of the cell cycle is the G2 phase, which is also known as the growth phase 2 or the gap phase 2. This is where the cell is growing and doing some more functions, which means it's expressing its genes, making its protein and doing what it needs to do in preparation for division. Which leads us to the M phase or mitosis. This is where the cell will divide into two identical daughter cells. It does this by the chromosomes separating at the centromere so that one of the original chromatids forms a chromosome in its own right in the new daughter cell. Similarly, the other chromatid that was present in the original parent cell will form its own chromosome in its own right in the other daughter cell. Each of those cells then will go back round through the cell cycle. The yellow sections of the cell cycle I've represented as the interphase. But if we focus on the DNA for a second, it's important to notice that sister chromatids are not always present within the cell. They are developed in the S phase, they are present in the G2 phase, and that's in preparation for the centromere splitting back so that the DNA actually only has a single piece of double-stranded DNA at the conclusion of mitosis and in the G1 phase.